Acetylcholine is the neurotransmitter that is released into the neuromuscular junction. Due to the release of this neurotransmitter, the chemically gated sodium channels open. This causes the muscle cell membrane to depolarize. The depolarization occurs along the T-tubules of the muscle cell membrane to release calcium from the sarcoplasmic reticulum into the muscle fiber. This phenomenon is known as excitation-contraction coupling.
The released calcium ions will then bind to troponin, which will change the tropomyosin and expose the crossbridge sites. The myosin heads are globular structures that will bind to the crossbridge sites to form a crossbridge. Next, ATP has to bind to the myosin heads, where it will be hydrolyzed into ADP, an inorganic phosphate. As a result, the myosin heads are in the energized state, and now they are ready to bind to their crossbridge sites. When the myosin heads bind to make the cross bridges, the phosphate groups break off. This causes the myosin heads to return to their former shape. And since the cross bridges are still intact, the entire actin filament moves along with them. This movement, called the power stroke, forces the filaments to contract.
After the movement, ADP is also detached, and a new ATP molecule can be hydrolyzed to energize the myosin head again. This completes one cycle of muscle contractions. Okay, let me clarify this. The next cycle begins when this ATP binds to the myosin head and breaks the cross bridge. Then ATP hydrolyzes into this ADP and this phosphate, which energizes the myosin head again. The phosphate breaks off, which binds the myosin head to the binding site. Then the power stroke contracts the filaments, and ADP is detached to be recycled again.